PCR invention. Carrie B. Mullis invented PCR and he got Nobel Prize for Medicine in 1993 for the invention of PCR. Now what is the principle of PCR? Let us see the principle of PCR. The target sequence of nucleic acid is denatured to single-stranded. First of all, the target sequence of nucleic acid is denatured to single strand. Then the separated strand is added to the reaction mix which contain primers, nucleotide, covalent ions, buffers and enzymes. Each strand is then allowed to hybridize with primers, and the process is known as annealing. The primer template duplex allows the enzyme DNA polymerase to add nucleotide to third terminus end of each primer and extend base sequence complementary to the target DNA sequences. This is known as extension. These three steps, that is renaturation and synthesis, are repeated again and again to generate millions of copies of target DNA. So I hope students you understood the principle. You will have to memorize it. Now requirement. What are the requirement? For target DNA, two primers, that is synthetic oligonucleotide of 70 to 13 nucleotide length, 4 deoxyribonucleotides DTP, DCTP, DGTP, DTTP, 4 or DNA tag polymerase that can withstand temperature up to 95 degree Celsius. Now what are the steps? The first step is extraction and denaturation. Here the nucleic acid is first extracted from the organism or a clinical sample by heat, chemical or enzymatic method. The target nucleic acid is added to the reaction mix which contain primers, nucleotides, covalent ions, buffers and enzyme DNA polymerase, and placed into a thermal cycler. On raising the temperature to about 95 degrees Celsius for about 1 minute, the DNA gets denatured and the two strands separated. Now here in the first step the two strands of DNA is separated. Now in second step, this is known as annealing. Here when the temperature drops to about 55 degrees Celsius, the two primers bind the two primers bind with the target DNA. That means at 55 degrees Celsius the two primers bind with the target DNA in their complementary flanking region. One primers bind to each strand. The two parental strands do not re-anneal with each other because the primers are in large excess over the parental DNA. Now the third step, extension. The new DNA strands are synthesized by Taq polymerase at 72 degrees Celsius for 30 seconds in presence of 4-deoxyribonucleotide. The new DNA strands are synthesized by Taq polymerase at 72 degrees Celsius for 30 minutes in presence of 4-deoxyribonucleotide. The primer template duplex allows the enzyme Taq polymerase to add nucleotide to the third prime terminus and of each primer and extend the base sequence complementary to target DNA. The step 1, 2 and 3 are repeated again and again. In each cycle the DNA strands are doubled, thus 20 cycles provide a million times amplification of target DNA. Now the fourth step is product analysis. Gel electrophoresis of the amplified product is commonly employed after amplification. The amplified DNA is electrophoretically migrated according to their molecular size by performing agarose gel electrophoresis. The amplified DNA forms clear bands which can be visualized under UV light. Here are the diagrammatic sketch. You can see the parent DNA molecule plus nucleotide. Just go through this diagram, focus it, and I'm sure you will be able to understand. Okay, thank you for now.